In this video, we're going to talk about one of our standard ASA monitors, capnography. Capnography, otherwise known as the measurement of our end-tidal carbon dioxide. So this is one of our most important monitors because it allows us to measure the expired carbon dioxide and basically track its waveform during the respiratory cycle. And that kind of helps us verify adequate ventilation and verify adequacy of ventilation and troubleshoot if we need to. So how does capnography work? We'll probably have two separate videos. We'll talk about kind of the physics of it here, and then we'll talk about what it looks like throughout the respiratory cycle in another video. But let's start by drawing. So we're going to demonstrate how this works. How does it work? And this is going to be a brief physics lecture here. So here we have our lungs. Here we have our end-tidal carbon dioxide sensor. So CO2 sensor. And this principle of how we measure the carbon dioxide concentration is based on the Beer-Lambert law of absorption. Law of absorption. And this law of absorption is also found in how we detect pulse ox, how we detect the oxygen saturation when we do pulse oximetry. So it's kind of it's a recurring theme here. So as our anesthetized patient exhales, which is going to be demonstrated by these arrows, we have carbon dioxide that is a specific concentration in our lungs. As our anesthetized patient exhales, that carbon dioxide concentration, represented by these dashes, will go into this device, this CO2 sensor. And this CO2 sensor has a light source, a light emitting source, which emits or shoots infrared light in its sensor. So infrared light through one end of the device, and then there's a base plate at this end of the device that is an absorber or a sensor. So you have an emission point and you have a sensing point. So as our patient exhales carbon dioxide, that concentration goes into this device. Infrared light shoots from one end to the other end and passes through this gas. Now, carbon dioxide absorbs infrared light. So as you can imagine, the higher the concentration of carbon dioxide that you have in this chamber, or the more that you breathe out from your lungs, the less light gets sensed, the less infrared light gets sensed at this end of our sensor. For example, if we shoot, let's just make this very arbitrary and simple. I just drew 4 arrows here. If we have 4 photons of infrared light that's emitted, and we have a concentration of carbon dioxide that absorbs a decent amount of that infrared light, proportional to the amount of CO2 that we have in here, then let's say 3 of these arrows are going to get absorbed by the carbon dioxide, and you're left with 1 arrow or 1 photon of infrared light. And this sensor is going to be analyzed using a graph, and that is going to reflect how our carbon dioxide concentration changes throughout the respiratory cycle. And we'll get into this part in another video.